Hello, grade 11. How are you today? Today we are going to talk about the genotype. In a species, the different alleles of a gene produce different phenotypes. Except for identical twins, experiments of tissue grafts showed that each individual is unique within his species. What is the reason of this uniqueness? Let's talk about the phenotype and genotype. The genotype is the whole set of genes or the combination of different alleles carried by an individual. Most living things are diploid and carry two copies or alleles of the same gene, each present on a homologous chromosome. The individual is called homozygote if the two alleles are identical, and heterozygote or hybrid if they are different. The allele that determines the phenotype is called dominant, and the other is called recessive. Therefore, the recessive alleles are only expressed if they are in the pure or homozygote state. So if we start with the genotypes as the genetic makeup of an individual, we can move to the phenotypes, which are the physical traits and characteristics of an individual and are influenced by their genotype and the environment. The ABO blood system is controlled by three genes, A, B, and O. A and B are codominant or they are expressed equally, and both are dominant over the allele O, which is recessive or masked. The rhesus factor is an inherited protein found on the surface of red blood cells. If your blood has this protein, you are Rh positive. If your blood lacks this protein, you are Rh negative. The gene for Rh positive is dominant over that for Rh negative. So, if an individual has the genotype AA or AO, then his phenotype would be A. If he has BB or BO, his phenotype would be B. If he has A and B at the same time in his genotype, then the phenotype would be AB. However, if he has the two alleles O in his genotype, then the phenotype is O. If he has RH plus or RH plus RH minus, then his phenotype would be RH plus. However, if he has both alleles RH minus in his genotype, his phenotype would be RH minus. Let's talk about the biological identity and genetic printing. Like a fingerprint that identifies an individual, nowadays it has become possible to trace his genetic print with a lot of certainty. In 1980, a scientist named Jeffrey developed this technique known as Jeffrey technique. In this technique, a blood sample is extracted and DNA is extracted from the blood cells. The DNA is cut into fragments by a restriction enzyme. The DNA fragments are separated into bands during electrophoresis in an agarose gel. The DNA band pattern in the gel is transformed to a nylon membrane by a technique known as southern blotting. Then the radioactive DNA probe is prepared. The DNA probe binds to specific DNA sequences on the membrane. Next, excessive DNA probe is washed off. At this stage, the radioactivity probe is bound to the DNA pattern on the membrane. X-ray film is placed next to the membrane to detect the radioactive pattern. And finally, the X-ray film is developed to make visible the pattern of bands, which is known as a DNA fingerprint. So by looking to this autoradiography and comparing it, we can conclude that individuals 3 and 10 are identical twins since they have identical banding pattern. And we can conclude also that each individual is genetically unique. Finally, I would like to thank you for your attention. Have a great day.